Okay, so the kind of data that we're focusing on for this talk is integrated biomedical data. And what I mean by that is we're taking clinical measurements from patients. So this could be demographic information, or it could be laboratory measurements. Combining this with kind of the insides of the patient, so this could be genomic data, proteomic data, metabolomic data. Integrating these together and trying to find relationships between both types of data. And our claim here in the causal discovery workshop, so that useful knowledge from this data requires some sort of causality. And I'll give two examples of sort of useful knowledge you can get from this data. So one is patient stratification, so trying to divide patients into different treatment groups based on their genetics. So you can do this without causality, but if you don't understand the causality underlying this sort of stratification, you don't know why your methods are working, and you don't know when they're going to stop working, more importantly. Another example is mechanistic analysis. So if we're trying to find how genes affect each other, or how genes affect other genes' protein products, this is inherently causal, meaning that if we try to knock out a certain gene and see how it affects the rest of the body, that is exactly what causality tries to capture. So why is this challenging? Well, it's because of some of the properties of this sort of integrated data. We tend to have very low sample size, so usually hundreds of samples is what we're looking at in a good case. We have many variables, so if you think of all of the genes in the human genome, that's over 20,000. If you try to incorporate clinical data and other types of omics data, this blows up really fast. We also have latent confounding in this data, which just means that we don't know that we've measured all of the variables that are relevant to our system. And in particular for causal analysis, this means we don't know if we've measured variables that affect at least two other variables causally in our data. And it's called the causal sufficiency assumption. And this data is mixed, right? So we have continuous data for sure from all of the omics measurements, as well as some of the clinical data. But most of the clinical data is also categorical, right? So if you think of demographic information or some sort of treatment or some sort of outcome, these things are often categorical. So in this talk, I'm just going to present some new strategies for causal discovery on this sort of integrated data. I'll provide a comparison of the benefits of each of the approaches on just simulated experiments. And then we have like a small sample biomedical data set that we applied one of the good performing strategies on. So first I'll give some background. So this is kind of, we talked about PC earlier. This is kind of the standard when you have latent variables. It's called FCI, or fast causal inference. I won't go into all the detail, but it's a sound and complete algorithm for structured learning in this case. And there's 5 major phases that you need to kind of understand. So it starts with an adjacency search, just like PC does, which we described before. And then you have to orient colliders. Colliders are those V structures that we talked about earlier, basically like the earthquake and the alarm, that sort of structure in the data. But then the difficulty with latent variables is that when you don't have latent variables, if two variables are independent, they're independent conditioned on some set of their neighbors. It's kind of hard to see, it's a subtle point, but it's not true if you have latent variables. Like, it could be independent on some other variable. It's actually not connected to the variables that you're talking about. And there's a whole theory about this. I can refer you to a book later if you're interested. But then we need to reorient these colliders and then make final orientations based on some constraints like acyclicity. So I'll briefly discuss collider orientation. So the general problem is we have this triple of variables. So we have A, C, and B here, where A and B, we're trying to find out if they both cause C. And basically how we do this is we look at the set that separates A and B. So A and B are conditionally independent given some set S. What we want to know is C in that set S? If it is, then we won't orient as a collider. Okay? If it's not, then we will orient as a collider. And this is the same reason as that earthquake, burglary, alarm example. But the question is, how do we actually identify the correct separating set or a probable separating set for a pair of variables? So there's been some prior strategies to do this. FCI and FCI stable, what they do is they use the first separating set they can find. So you start by conditioning on 0 variables, you slowly increase this number until you find some set that makes those two variables conditionally independent. And that's going to be our true separating set. The issue is in small sample size, this is not always reliable to orient these colliders in this way. So there's been some adaptations to try to fix this problem. Conservative FCI is very conservative about this. So it actually tests all possible separating sets and makes sure that none of them contain the middle variable. If that's true, then they orient as a collider. If all of them do contain the middle variable, then it orients as a non-collider. It's definitely not a collider. Otherwise, it says it's not sure, and it just leaves it as undirected. Another possibility is to use a majority vote. So you could look at all these sets again and just take a majority vote as to whether that middle variable should be included in the set or not. For this comparison, we focus on CFCI because it historically has performed the best in these comparison studies. And we also use just FCI stable as a baseline. But the strategy we're going to propose is this max strategy that was recently proposed last year. And basically what we do is we choose the separating set that has the maximum p-value in its conditional independence test, and we use that as the true separating set. And these distributions can give you a pretty good idea as to why. So what we're trying to do is find a set that's least likely to imply dependence, right? And dependence is the alternative hypothesis, is H1 in our case, which is the distributions on the right. So if we want to be least likely to be in one of these distributions, we would look to the tail, which is at the end, right? So the maximum p-value, this is kind of intuitive, should be, is least likely to be in one of these distributions, is more likely to be in the uniform distribution. Okay. The issue is, again, when we're doing this max search, we need to test all possible subsets of the adjacencies of our variables, which blows up pretty quickly. So what we try to do is make this faster by constraining the adjacency search at the beginning so that we have less subsets to test on. We do this with an undirected graph learning method. So first we learn an undirected graph on all the variables. So normally FCI and PC start with a fully connected graph. Instead of that, we use this constrained undirected graph as our beginning. So this prevents the necessity of this full adjacency search, and instead we can start with this undirected graph. But the nice thing is we retain this asymptotic correctness if the learned undirected graph is a superset of the true adjacencies. And so this naturally gives you a trade-off. So you can try to delete as many edges as possible, where you might lose correctness. You might delete an edge you were supposed to keep. Or you can delete very few edges, in which case you'll hopefully still be correct, but your algorithm's going to be slower. It's going to take longer. And so what we propose to learn the undirected graph is this algorithm called MGM from Lee and Hasty in 2013, so mixed graphical models. And here's the joint distribution. It's a Markov random field on mixed variables. I won't go into this detail, but basically the x's are your continuous variables. So there's p continuous variables in this example. And there's q categorical variables, which are your y's. And so each term is just the interactions between, first, both continuous variables, continuous node potential, the mixed edge potential, and then just discrete edge potential. And the nice thing about this joint is if you look at the conditional distributions, they're nice and easy to work with. So a conditional distribution of your categorical variables is a multinomial distribution. And the conditional distribution of your continuous variables is Gaussian in this case. And so to actually learn the MGM, we minimize the negative log pseudo likelihood using a proximal gradient approach. In the original MGM paper, they just have one sparsity penalty. So they use one lambda to regularize this model. What we found in 2016 was that actually splitting this by edge type, so having one regularization parameter for each type of edge, continuous, mixed, and discrete, actually improves the performance of the model. So this is what we do in this case. So finally, our independence test. So this is actually was mentioned earlier too, is like the fast test. But basically, we do a linear regression if everything's continuous. And we do a likelihood ratio test if anything is categorical. If either X or Y is categorical, we do a likelihood ratio test on logistic regressions. Okay, so those are our proposed methods. I'll just go through some simulated results now. So first, here are the competitors again. So we have FCI stable, FCI, which is just abbreviated to FCI for the rest of this talk. We have conservative FCI or CFCI. FCI with the max strategy is going to be FCI max. And then if we combine MGM with any of these algorithms, we just put MGM at the front. So I know there's a lot of abbreviations, but that's how we write them. And then finally, this is one competitor externally, is BCCD, Bayesian Constraint-Based Causal Discovery. The main idea there is they also can handle latent variables on mixed data. They do it in a Bayesian way. So I won't go into those details, but it's referenced in the paper if you look at it online. And then our output metrics are precision and recall. We do this for adjacencies and for orientations, which I'm going to discuss in a second, because it's a little bit different on latent variable graphs. We also check the running time of the algorithms. Okay, so this is how we compute whether our orientations are correct. So I'll just quickly go over what these endpoints mean. So if you look at just the column headings, we'll start on the right side. So when you have a tail, that's a prediction of an ancestry relationship. So that means, in this case, B is an ancestor of A. When you have an arrowhead, that's a prediction of a non-ancestry relationship. So in this case, B is not an ancestor of A. Okay. If you have a circle, that's we don't know. So it could be an arrowhead or it could be a tail. It's not invariant in this equivalence class. Okay? And you're learning an equivalence class of graphs. Okay. So how we score this is if our endpoints are correct, we call it a true positive. If we predict a claim of ancestry wrong, so if we predict that A is an ancestor of B and we're wrong, we call that a false positive. If we predict a claim of non-ancestry wrong, so if we predict A is not an ancestor of B, and we get it wrong, we call it a false negative. Yeah?
So by ancestor, it can be anywhere in the graph as long as it's an ancestor.
Correct.
So it's not direct.
Right. Yeah, and the reason we call it an ancestor is you imagine just we have a 3-variable graph, A causes B, B causes C. If B is latent, there's no way to know that A wasn't directly causing C, right? Just if you think about that, there's just no way to figure that out. Unless you make very specific assumptions about how this works. So that's how we compute the score. This is what we're going to use to compare the algorithms. So here's our results on 50 variables and 1,000 samples. So I'll just explain how these graphs work. So each column is a different edge type. So on the left, we have edges between continuous and continuous variables. The middle is mixed variables. And discrete variables are on the right. And then in the top row, we're comparing adjacencies. In the bottom row, we're comparing orientations. And then each individual graph, the x-axis is precision, and the y-axis is recall. So ideally, you want to be in that top right corner in each graph, and that means you're doing a good job. So our main takeaways from this is that BCCD does really well on continuous adjacencies, but not well when you incorporate mixed data at all. In terms of orientations, we actually find that this FCI max strategy does the best in balancing the two. We see that FCI has low recall, which again in this case means that it's trying to orient a lot of arrowheads, making lots of claims of non-ancestry. CFCI tends to have high recall and low precision, meaning it's not orienting any arrowheads, which if you go back to what it does, remember it's very conservative about orienting colliders, which is reflected here. Right? We have very few orientations of colliders, which is how you get arrowheads. And yeah, so those are the takeaways we got from this. So then we incorporated MGM into each of these strategies. So we left max as is in the red. And then we have MGM FCI, MGM with CFCI. And then MFM is MGM with FCI max. Writing that all out is a little cumbersome. So we abbreviated that to MFM. So our main takeaways from here is that MGM FCI Max provides us just a slight orientation advantage over FCI Max alone, but it's almost, you can't even see it in these particular graphs. Both approaches perform better than the other MGMs, so MGM with CFCI and with FCI. But again, these have better recall, so it just means they're more conservative in orienting colliders. So then we scaled this to 500 variables, and this is closer to what our actual biomedical data looks like, if you remember at the beginning of the talk. And fewer samples. This is 500 samples only. And so here what we find is that MGM FCI Max actually does really well in orientation again. The only downside is this discrete adjacencies. So it's actually very parameter dependent. So the sparsity parameters I talked about earlier, if you choose them incorrectly, you keep the same precision, so you're only getting correct edges, but you're getting much fewer of them. So there's a trade-off there in trying to figure out exactly what lambdas to pick in MGM. Oh, and I should have mentioned this earlier, but each point here is just a different setting of the parameters. So for each algorithm, each point is a different setting of the parameters. That way we can explore how sensitive it is to different settings of the parameters. Okay. And then finally, the efficiency. So this is on 500 node graphs again. And what we're looking at here is on the left, runtime versus alpha. Alpha is our cutoff for the conditional independence tests. And what we find here is that with like a standard setting of alpha is usually 0.05. That's what most people take to be an appropriate setting. That's all the way on the right in the graph on the left. So if you look at that, FCI is by far the slowest when you try to increase the alpha because it's not able to delete many edges in its adjacency search. Whereas the MGM methods do that a priori, so this like normal setting of alpha doesn't really hurt. But if you use a very low setting of alpha, like 0.01, a very strict threshold, then FCI and MGM tend to be comparable. And the right's just illustrating this. This is the independence tests performed. And this is why MGM can be faster, is if you get a huge savings on the number of independence tests performed, you get a faster result. So finally, we applied this to a biomedical data set. This is definitely smaller than the data sets we were analyzing in the lab, but just as an example. So this is to study the effect of HIV on lung function decline. So after filtering out some of the variables and samples with missing data, we get a final data set of 14 continuous variables, 29 categorical, and 400 samples. So first, we just have some edges that we found to be encouraging, just as that they make sense. So smoking related to coughing, related to having a sore throat, related to having digestion is something we'd like to see. Chemotherapy linked to cancer is something we'd like to see. And then height, weight, BMI, and gender. And then we have some new associations also. So the important one here is the CD4 to DLCOPP final. So that DLCOPP is just a measure of lung function. CD4 is a measure of immune system decline. It's the CD4 cell count. And actually recently this has been found to be correlated, which we also found, but this provides some evidence that the relationship might be causal. So it might be that having a low CD4 cell count could affect your lung function. So here are the final conclusions. The MAX strategy improves orientations by balancing precision and recall a little bit better than the other strategies, which again means just balancing orienting too many colliders with not enough colliders. However, it's too slow to be used in practice without some optimizations, which we saw. So when you do the MAX strategy again, you're trying to condition on all subsets of variables, which if you don't have some optimization in place, that takes too long, especially on data sets above 50 variables. MGM allows this MAX strategy to scale a little bit better to large numbers of variables on mixed data. Our orientations definitely still need to be improved on real data, but usually adjacencies are highly reliable. You don't find all of the adjacencies, but if you saw on those graphs, precision was close to 1 almost always. So everything you find is very reliable. For future directions, we definitely, in general in causal discovery, we need more widespread simulation studies with data generated from several different distributions to actually determine what methods are useful in different situations. This we tried to do here, but this is a very small sampling of that. There's a lot more that needs to be done. Where we plan to use this type of method in a biological case is to look at microRNA, mRNA interactions. So what we want to find is mRNAs that are actually confounded and see if we can find a microRNA that can target both of them. But before that, we need to determine whether the latent confounders themselves, finding where there are latent confounders in the graph, is that part accurate? So that's a more specific look at the results of this data. And then finally, you guys saw that we were ranging across parameters before in those results. So the question still remains on how to pick the appropriate parameter for your data set. We have some methods that try to find the most stable parameter, but this is still an open problem on most causal discovery methods. So finally, I'd like to acknowledge my sponsors, Dr. Benos, Dr. Chrysanthus, Dr. Morris from UPMC for the biomedical data, and then everybody from CMU Department of Philosophy for their help on the causal modeling algorithms. And then lastly, I have a shameless advertisement from my advisor that the lab needs some postdocs. So if you're interested in this sort of causal graphical modeling and applying it to big biomedical data, that's what we do. And then usually to some sort of cancer data is the final goal. And you can send an email there. All right. Thank you, everybody.
Yes, thank you.